So welcome to the final lecture on the retina. And in this lecture, we're going to just briefly talk about retinal circuits. So in previous lectures, we mentioned the vertical information flow through the retina. That's the photoreceptors to the bipolar cells to the retinal ganglion cells. So we're first going to talk about that. And we've already talked about the photoreceptors, the rods and the cones. Now let's talk about this next layer of the retina, the bipolar cells. There are many types of cone bipolar cells. Those are the bipolar cells that get the glutamate from cone photoreceptors. And there is one type of rod bipolar cell. And so the numbers vary a little bit based on species, but this is for humans. There's a whole range of both off cone bipolar cells and on cone bipolar cells. And then the one rod bipolar cell type is an on type. And so what this means is when you decrease light, these off cells depolarize. When you increase light, these on cells depolarize. So we were talking previously about how photoreceptors hyperpolarize in response to light. So they would be like an off cell. So all these off bipolar cells, they express AMPA or kainate glutamate receptors. So these are ionic channels. So when the photoreceptors release glutamate onto these cells, they are sign preserving, as in hyperpolarization of the photoreceptor causes a hyperpolarization of these off bipolar cells, and vice versa. A depolarization of the photoreceptor causes a depolarization of these bipolar cells. So they respond in the same way as the photoreceptors. But these on bipolar cell types, they all express a metabotropic glutamate receptor, specifically mGluR6. And so this is a little more complicated than the ionotropic receptors, and it will invert the sign. So actually when glutamate binds to this receptor, it starts a cascade that results in less depolarization. So you have an off photoreceptor that gets inverted to an on bipolar cell. So they're still receiving glutamate from the photoreceptors, but they're switching the sign. And now when the photoreceptor hyperpolarizes to light, these bipolar cells depolarize to light. So for your first summary question, I'd like you to answer, if you took the rod bipolar cell and placed all of its mGluR6 receptors with AMPA receptors, how would the cell respond when you increased the light intensity onto the retina? So you can broadly split them into off and on cell types, but there are finer distinctions, which would make sense. You don't want to have a whole bunch of different types that all respond in exactly the same way. And there's various distinctions, but one of the simplest way you can categorize them is that they some are more sustained and some are more transient. And they generally stratify based on this sustainedness and transientness. They also, I should say, stratify based on the on and off. So you'll see they all collect glutamate from the photoreceptors, but their axons terminate in different layers of the inner plexiform layer, different sublayers of the inner plexiform layer. So all these off bipolar cells stratify in the off layer of the inner plexiform layer, and then all of the on bipolar cells stratify in the on layer of the inner plexiform layer. And then generally in the middle, near the boundary between on and off, you find more transient stratifications. And then towards the edges, you find more sustained stratifications. So the rod bipolar cell is an on cell, stratifies in the on, and it's pretty sustained in its response. So this makes it pretty simple for a ganglion cell to collect the kind of information it wants. So if you want to be an off retinal ganglion cell, you send your dendrites to the off layer and collect from off bipolar cells. If you want to be an on retinal ganglion cell, you send your dendrites to the on layer, and you can pick more sustained or more transient. And so we show here on and off bipolar cells, or sorry, on and off retinal ganglion cells, just like there's on and off bipolar cells, but there's also more types of retinal ganglion cells. So I mentioned earlier that you just get this greater and greater diversity as you proceed through the layers. So there's on, there's off, there's on-off, and it's a little unsure what all is in humans and what's just in mice and other lower vertebrates, but definitely in mice, you have a huge explosion. You have cells that are sensitive only to movement. You have retinal ganglion cells that are only sensitive to movement in a specific direction. So they'll like spike when a bar moves by on the left, but if it goes to the right, they don't do anything. So you get these very specific types of responses from the retinal ganglion cells. So let's go ahead and then walk all the way through the pathway. And how do the cells respond to either a light spot, so an increase in light shown on the center, or a dark spot shown on the center? So if you increase the light level, you shine a light spot on a cone, it will hyperpolarize. It will then release glutamate onto both on and off bipolar cells. The on bipolar cell will invert that sign, and now instead of hyperpolarizing, it will depolarize. The off bipolar cell will keep the same sign, and it will hyperpolarize also. And then if you have an on ganglion cell, it will be connecting to the on bipolar cell, and it'll increase its spiking. If you have an off retinal ganglion cell, it will decrease its spiking. And everything's the exact opposite if you shine a dark spot onto the cell. But this brings up the concept of center and surround. So these stimuli were only shining in the center, right over top of the photoreceptor. But to the sides of the photoreceptor, beyond the photoreceptor, is what we call the surround. This is just text describing what I just said. But so that area surrounding the center, the surround, it can also influence the response of the cells. And usually the surround is suppressive, as in it inhibits. So here is a retinal ganglion cell with surround suppression. And if you shine a light right in its center of its receptive field, it increases its spiking. So this is an on-center ganglion cell. And then if you do it to an off-center ganglion cell, it decreases its spiking. And with a dark spot, you see the opposite. The on cell decreases spiking, the off cell increases spiking. But now what they've done is they've included, they first go center only, like we saw previously, and then they include more light in the surround. And what you see is these cells, the on cell decreases its spiking. It was spiking a lot, and then adding more light to the surround decreases its spiking. And the off is the opposite. It decreased its spiking a lot to the on stimulus. Now it increases. So basically, the surround undoes some of the response to the center. It suppresses that response. So what is the molecular basis of this, or what is the circuitry basis of this? And it comes down to that we've been talking about the vertical information flow, but there's also the horizontal information flow, if you remember from the previous lecture. And that's lateral inhibition provided by horizontal cells and amacrine cells. So here we're just showing the horizontal cells. And what you can have is cones in the surround excite a horizontal cell, which then inhibits the center. So if you don't have anything in the surround, you don't have inhibition. Once you start shining light in the surround, the horizontal cell is able to inhibit the center. And the same thing can happen with an amacrine cell. It would just happen at this synapse here between the bipolar cell and the ganglion cell. So for my second summary question, I want to ask, which visual stimuli would cause the most spikes for an off retinal ganglion cell with strong surround suppression? So tell me, would you want to increase or decrease light in both the center and the surround? What would give the most spikes for an off retinal ganglion cell with strong surround suppression? So one last point to talk about is we've been talking about the vertical pathway of cones, and it's a little bit different with rods. So the rod pathway is weird because rod bipolar cells don't connect directly onto retinal ganglion cells. They don't release any glutamate onto retinal ganglion cells. They kind of hijack the cone pathway. So there's 2 or 3 different ways that they can do this. The primary way they do this is the rods excite the rod bipolar cells. The rod bipolar cell then releases glutamate onto an A2 amacrine cell. So this is an amacrine cell that it's a very narrow field amacrine cell, so it extends a little bit laterally. And so this A2 amacrine cell is then excited, is depolarized when light turns on. And then it makes a gap junction, an electrical synapse to an on bipolar cell, and then the on bipolar cell releases glutamate onto an on ganglion cell. So it just kind of works through an A2 amacrine cell as a bridge to connect up with an on bipolar cell. So that's the primary rod-on pathway. But what if you want to provide input to an off ganglion cell? So then there's the primary rod-off pathway. So the primary rod-off pathway starts with the same components. A rod photoreceptor synapses onto a rod bipolar cell. The rod bipolar cell synapses to an A2 amacrine cell. But now things are different. The A2 amacrine cell forms traditional synapses with an off cone bipolar cell, and it releases glycine, which is inhibitory. So the A2 amacrine cell inhibits the off cone bipolar cell, which then forms a traditional glutamatergic synapse to the off ganglion cell. So it's pretty much the same as the on pathway, except that we have one more sign inverting synapse. So if you shine a spot of light onto the cell, you would see hyperpolarization inverted to depolarization, maintained as depolarization, inverted to hyperpolarization, maintained as hyperpolarization. So you can kind of count up how many negatives you have. In this setup, you only have 1 negative. In this setup, you have 2 negatives. So you revert back to off. There is a secondary pathway, which I think is simpler, and that is you can just simply have the rod form a gap junction with a cone. So at this point, if it's dark out, the cone wouldn't be responding, but the electrical synapse from the rod to the cone allows some of its hyperpolarization to light to leak through into the cone, and then it's just the normal cone pathway all the way down. So this is the exact same between on and off. You just hijack into a cone at the very earliest stage. And then there is a tertiary pathway for off only that might play some role in that maybe rod bipolar cells do synapse some directly onto off cone bipolar cells. So for the third and final summary question, I want you to answer: if you were to delete the genes necessary for forming gap junctions in the A2 amacrine cell and in the rod photoreceptor, your vision would be impaired most in which light level? Scotopic, mesopic, or photopic? And then this last slide is just summarize, or these last two slides are just summarizing what I just said verbally.